So in section 2, we're going to look at the form and function of the eukaryotic cell. So we're looking at the external structures and boundary structures. So we're starting from the outside and going in. And so we're going to be looking at the flagellar structures of bacteria, archaea, and eukaryotes and be able to differentiate between those. Probably won't be able to do that until we actually talk about the bacteria and archaea. So that will come back later. Another outcome would be the characteristics of Glycocalyx in eukaryotes and the eukaryotic microorganisms, which eukaryotic microorganisms might have a cell wall, and then list the similarities between the eukaryote and bacterial plasma membrane. So again, because we're doing this in the opposite, we may not be able to compare them until later, but those are the outcomes. So some of the features that we see in eukaryotic cells, that are found in all eukaryotic cells, are the plasma membrane. That's found in all cells, period. And then a nucleus, which is unique to eukaryotic cells, mitochondria, which is unique to eukaryotic cells, endoplasmic reticulum, Golgi apparatus, vacuoles, cytoskeleton, and glycocalyx. So we see this is eukaryotic cells have membrane-bound organelles, which include these endoplasmic reticulum, mitochondria, Golgi apparatus, vacuoles. Those are all unique to, and the nucleus, they're all unique to eukaryotic cells. They're not found in bacteria. They're not found in archaea. Cytoskeleton is actually found sometimes in bacteria, and glycocalyx is also sometimes found in bacteria. So, but these structures here, the nucleus to the vacuoles, those are all membrane-bound organelles. And like I said, cytoplasm is found in all cells. And then in some eukaryotes, we have cell wall, locomotor appendages, and chloroplasts. So here is a picture of a typical eukaryotic cell. And you will never see a cell like this because this has structures that are unique to bacteria, plants and animals, or cells that have a cell wall and cells that don't have a cell wall, and cells that have chloroplasts and cells that don't have chloroplasts. So like centrioles here, those are unique to animal or animal-like cells, and chloroplasts are found in plant or plant-like cells, and those are not found usually in the same cell. The rest of these structures are pretty typical. Like you're going to find them in most of the cells. In all eukaryotic cells, you'll see this yellowish orange color. They're found in all eukaryotic cells, but these ones in red, the cell wall, the glycocalyx, the centrioles, the chloroplast, the flagellum, those are all, found in some eukaryotes, but not all. So the different structures, we're going to look at the nucleus, which is in purple here, with the dark purple or the round center here that is called the nucleolus. The nuclear envelope is double membrane, and it is continuous with the endoplasmic reticulum, which this is the endoplasmic reticulum with ribosomes, so that is rough endoplasmic reticulum. And then there's smooth endoplasmic reticulum, which does not have ribosomes on it. And they have different functions, so we'll go over that. And then mitochondria that you can see here has lots of folds in it and has a double membrane. The chloroplasts that have a double membrane and have these stack of green disc shapes, and those are unique to photosynthetic organisms. Lysosome, so like a bigger vacuole that comes off, or a vesicle that comes off of the Golgi apparatus. So you can see it's shown in orange, and the vesicles that come off of that are also in orange. There's also these transition vesicles that go from the rough endoplasmic reticulum that then fuse with the Golgi apparatus. And then you can see throughout, you can see all these different cytoskeletal structures. Some are thin, some are hollow and thick. And then you have the flagella that is, again, not found in all cells. So the flagella, we'll start with the external appendages. These extend out of the cell. They actually take some of the cell membrane with them. So the extension is covered by the cell membrane. It's thicker than the bacterial flagella. It is more complex, and it is covered by the cell membrane. That, in comparison to the bacteria flagella, is thinner. The bacteria cell or flagella is less complex, and it is not covered by the cell membrane. And then the eukaryotic cell has a long sheathed cylinder containing regularly spaced hollow microtubules in this 9n+2 arrangement, which we'll show you here in just a minute. So those are some differences, and you'll see then when we look at the bacterial flagella in chapter 4 how it's different. So here you can see the cell membrane covering it, and then it has these 9 tubes of dimer tubes of microtubules, and then one in the center. So it's the 9+2 in the center. So there's the microtubules running the length of the flagella. So this structural arrangement allows them to have different kinds of linear motion. It acts like a propeller and can bend and sway in different directions because of the connections between all of these, which you really can't see in this picture. The cilia are shorter, and they are more numerous, and they can be, several thousands of cilia, where flagella you may have a few or even just one flagella. Cilia are more numerous, and these are only found in protozoa. So this is something that I get students who miss this on every test. Something that is unique to protozoa is the cilia. We don't see these in bacteria. There are no cilia in bacteria. And there are cilia in our eukaryotic, or in our epithelial tissues, like cilia are found in your respiratory tract. And so that is true, but as far as the microorganisms that are concerned, this is really the protozoas are the only group. So certain animal cells and then protozoa are the only ones. So this is a very animal-like structure. You don't see it in algae. You don't see it in bacteria. It's just protozoa, and like I said, like our ciliated pseudostratified epithelial cells in humans and some animals, but just certain cell types. So they function in feeding. They kind of, as you can see, this the little cilia are kind of like hair-like structures here on this little stentor, and they kind of create this tunnel, a kind of vortex that forces the food into the gullet, which is where they absorb and take in the food. And so that's a feeding structure. And usually they're beating just to go toward a particular food source, but also to then take in the food. So that's their purpose. So the glycocalyx is the outermost boundary that comes into direct contact with the environment. So the appendages we just talked about extend from the boundary, but this is the outermost boundary. And it is called the extracellular matrix. That's probably the term you used in anatomy and physiology. So it is the layer of fibers and network of these fibers composed of polysaccharides that surrounds the eukaryotic cells and allows those cells, if it's multicellular, to attach to each other and form tissues and things. But it is the external connection to the cell. And in bacteria, it's much more defined. It is not as defined in eukaryotic cells. They do sometimes have a capsule. It's more like a loose network of fibers, a slime layer, but it's kind of more common in bacteria to have a capsule. So a cell wall in bacteria, they do have one. In eukaryotes, the only groups of eukaryotes that have it are fungi and algae, and they have a different consistency. So fungi have chitin, which is something that is and they can have cellulose, but chitin is something that's found in animals In crustaceans, for example, they'll have this as their exoskeleton. And so this is kind of an animal-like thing. So they have this layer of chitin and mixed glycans, which are sugars, and glycoprotein. So it's a very complex cell wall. And then they have the glycocalyx, or the outer part is the glycocalyx, and then you have the cell wall, and then the cell membrane, and then the inside of the cell. So that is usually the order. That's something you should keep in mind. From the outside, the outermost boundary is the glycocalyx, and then you have the cell wall, and then you have the cell membrane. And so it's made up of chitin and mixed glycans and glycoproteins, so sugars, polysaccharides, basically. But different than usually the algae. The algae are typically cellulose. And that is different than bacteria cell walls, which consist of peptidoglycans. So they have a different composition than the bacteria cell walls. So the cytoplasmic membrane is a layer of phospholipids, and every membrane-bound organelle is also going to be a layer of phospholipids. That's why it's called a membrane-bound organelle. And it's embedded with protein molecules, and some, particularly those that don't have a cell wall, will contain sterols like cholesterol, and that stabilizes the membrane. Otherwise, it would be just about the consistency of olive oil. So very much the proteins and the sterols help to stabilize the membrane. And it's a selectively permeable barrier. So what its function is to transport substances in, nutrients in particular that are needed by the cell, and to transport waste products out and other products that the cell may be producing. So concept check for section 5.2. Describe 3 ways in which the eukaryotic flagella are different than the prokaryotic flagella. What are the purposes of the cilia, and which organisms have cilia? What are the functions, the 3 functions of the glycocalyx, and differentiate between eukaryotic and prokaryotic cell wall structures. Which eukaryotes have a cell wall? So the 3 ways that the eukaryotic flagella are different than the prokaryotic flagella. One would be that they are bigger and they're more complex than the prokaryotes, and they extend with the cell membrane covering it. So in bacteria, the prokaryotic flagella, they are smaller, they are less complex, and they do not extend. They extend out of the cell wall and cell membrane, so they are not covered by a layer of the cell membrane. The purposes of cilia, it's for feeding mainly, but they can move toward what food sources they are looking for. And which organisms have cilia? Remember that only protozoans, those animal-like one-celled organisms, have cilia, and certain animal cells, like our body tissues, have cilia. Some of them, like our respiratory tract, has the mucus layer, the mucus escalator, that will help to beat upward and get rid of any kind of microbes that might get trapped, or dust particles that might get trapped in the mucus, and then we swallow it. We'll talk about that as one of our innate defenses in chapter 14. The 3 functions of the glycocalyx would be protection from drying out, protection from the immune cells, if it is a microorganism that has glycocalyx, and then to attach tissues. It's a connection and communication between cells. So we see this both in bacteria and eukaryotes, but in eukaryotes it can be a way to join cells and into tissues. So differentiate between prokaryote and eukaryotic cell walls. Their structure is made of different chemical composition. They're both kind of a glycoprotein kind of thing. The eukaryotes have cellulose, though, and chitin, which are pretty much repeating subunits of glucose, and not a lot of protein, where the prokaryote cells have peptidoglycan, which has some amino acids combined with a chain of carbohydrates. So it's a peptido and glycan. Glyco refers to sugar, and again, peptido refers to the protein, so peptidoglycan. Which eukaryotes have a cell wall? So plants and algae, those are two particular ones. They have cellulose cell walls, and then fungi. So that is section 2.